Hey folks, so recording single neurons is one of the coolest things we do in neuroscience and coolest things we do in science, period. It's one of the reasons that I got into neuroscience just because I thought this technique was so fascinating. Um, and I really think it's important if you're going to be successful in science to really like the techniques you're using. So I'm going to talk a little bit about recording single neurons in a specific technique called whole cell neuron recordings. All right. So to do this, you have, let's say there's a neuron you want to record. You have a glass micropipette and it's filled with an isotonic intracellular solution. So a solution that's similar to the internal composition of a neuron. And in that glass pipette, you have a recording electrode, and there's also a reference electrode in your solution. So as you get your micropipette very close to a neuron, you start doing these voltage steps with your electrode and measuring the current generated. And if there's a free flow of ions between your recording pipette and the reference electrode, you're going to get a one-to-one correlation between the voltage you change and the current that you record, right? But as you get really close to a neuron, those ions have trouble getting out from between this little space. So as you get really close, the current generated from each voltage step actually goes down. And that's one way that you can tell you're really close to a neuron, even if you're not able to visualize it with a microscope, which sometimes you can. So you get your recording pipette really close using a micro manipulator, so you can move microns at a time closer to these neurons. And as you get really close to it and the current generated from each voltage step gets really small, you then actually apply a little bit of suction and your glass pipette seals to the actual cell membrane so that there's essentially no flow of ions from your intracellular recording pipette out to your reference electrode. This is called a gigaseal. So you're doing your voltage steps, but you're getting no current generated. Okay? At that point, so you're in the attached patch mode, and you then apply a little bit more suction, and you actually rupture the little circle of membrane that is at the tip of your recording pipette so that there is free flow of ions of your intracellular solution in your micropipette and the inside of the neuron. And at that point in time, you also see a change in the current generated for each voltage step, because now you get this big curve because the cell membrane actually holds charge and discharges it. So it's and you can watch this happening on a computer or an oscilloscope as you're doing this experiment. So you can tell, oh, I formed this seal because I'm getting no response in my current, and then, oh, boom, I've ruptured and I'm now in the whole cell configuration. I have a continuous solution between my recording electrode and the inside of this neuron. And it's at that point in time that you do your experiment, right? You listen to either the neuron, doing their action potential. You can also hear what other neurons are saying to your neuron. They'll synapse on your neuron and cause little changes in current by their neurotransmitters binding and opening little channels in the neuronal membrane. And so that's how you record from a single neuron using whole cell recordings. I just think this is one of the coolest techniques out there, and it's just so great. I am going to start posting these little science lectures on YouTube as well. I've already started. If you want to learn more about neuroscience and how we record neurons. So hope you learned something.